So, thanks so much for coming today. We're so pleased to welcome Michael Long with us, and Ian's going to introduce him.
So, thanks everybody for coming. This is going to be an awesome talk. I'm really excited about it. So Michael is the Thompson Susanne Murphy Professor of Neuroscience and the Vice Chair for Education at NYU School of Medicine. He did his graduate studies with Barry Connors at Brown, where he investigated the role of electrical synapses in the mammalian brain. And then his postdoc work at MIT with Michale Fee, he started studying the songbird model to uncover the cellular and network properties that give rise to learnt vocal sequences. And we'll hear about this today. So he started his lab at NYU in 2010 and is focusing on the neural circuits underlying skilled movements, often in the service of vocal interactions. And you can see his lab just does some of the most amazing work. And he's also won lots of awards and funding, and great to have you here.
Thanks. Good. Well, thank you all for coming here. There's a good buffer zone here. I feel like different countries sit in different places. I was in England, they sit way in the back, and then I guess you are too. So today I wanted to boil down, this was a longer title for a while, and now it's just how parrots speak. And so I think we're getting close. We've worked with parrots now for about 2 years, and I think they're incredible, these budgerigars. And the motivation of this is really, I work at a medical center, and this is a patient that came into our medical center with expressive aphasia. So this Broca's aphasia, and it's probably one of the first things that we hear about in our neuroscience classes. And this person has agreed to be interviewed by a neurologist named Jonathan Howard. And you can see there's a clear block in his ability to really summon and then deploy words other than just basic yes and no.
And what happened?
Yeah.
You're not sure?
Yeah.
Did you have a stroke?
Yeah.
Okay. And what symptoms did you have? What's wrong with you now? Okay, so you can't move the right side of your body. Are you able to speak okay?
Well, yeah.
Okay. Do you feel that your speaking is normal?
No.
What did you used to do for a living?
Yeah.
Can you talk about it? You're pointing to the computer. I guess you work with computers, is that right?
Yeah.
Just talk about it a little bit if you can.
No.
You can't?
Yeah.
Does it bother you having trouble speaking?
Yeah.
So speech is actually quite difficult. It's an act that requires us to marshal about 100 different muscles and to have rapidly changing acoustic structure to hit that on about a 20 millisecond time precision. And of course, we don't just speak into the void. We have typically a conversation partner or many conversation partners at any time. And so this is just an annotated podcast where two people are talking for 2 minutes. You can see when this person is talking with this shape in white and then this person in gray. And what you can see here is this kind of back and forth turn taking. And the turn taking is something that's just a hallmark of conversation. And we've studied how the brain can do this. It's typically doing that because of an intense amount of planning. We summon a concept, we have it ready in working memory, and we could deploy it literally in a blink of an eye. Typically, this turn-taking time is about 200 milliseconds, so incredibly fast computation to do some very high-level cognitive, sensory, and motor acts. So the problem is about 15% of the population has communication disorders. And our lab and others have been recording from the human brain. We do intracranial recordings even at the level of neuropixels, looking at hundreds of neurons at a time. But that's tough and very limited, and we can only do in very specific cases, obviously. And so how do we model language? Well, language is unique to humans, but we think that there are specific aspects that can be modeled in these creatures. So in addition to our interests working at the medical center and with the University of Iowa, which is a long-term collaborator, looking at the human brain, doing more cognitive acts, we also have a whole group of animals that interact vocally, and we're understanding how their brain does this. And so one animal that's been, I guess, our longest running interest is the zebrafinch. And so we've had many people through the years that have worked on this model. And for those who know the zebrafinch, it's a learned act. And so less than 1% of the animals that exist on this planet can actually learn through vocal imitation. So this is quite a thing. And these guys learn their courtship song. So this is one thing that they can take and then present to a female, and then typically as part of the mating process. So this is the male. This is his son, and he's going to be teaching the song to his son. He uses some tough love here. And so the appeal of this, I think, is twofold. First of all, we have a really highly precise and quantitative, tractable behavior that we can see unfold over time. And so it takes about 200,000 or so practice trials to get this song perfectly right. And so this is what the tutor song looks like in a very specific experiment that Ofer Chernikovsky did many years ago. And what Ofer did is he took this kind of artificial robot bird, if you like, and has this bird wired up for sound. So this is a speaker coming out of the belly. And that bird can make this sound over and over. Let me just play that sound. Fine. That's an adult zebrafinch whose notes are advantageous for us to track for one reason or another, unrelated to this bird. But then after being exposed to this bird is raised with the mother. They can keep that bird alive and happy, but we've actually pulled that father out quite early. So the only sensory experience for the song, because the females don't sing, is this robot bird. And so if the bird is hearing this, you can see he's quite far away here. So if the adult sounds like this, the juvenile is like this. Kind of scratchy, right? But over time, I'm going to give you a kind of time-lapse movie, if you like. Again, they're singing all day long, and now I'm giving you an example every 5 days. And you see the blooming of this song, like a rose, and this looks just like the tutor song after about 4 months, right? So they're practicing, and that this one thing they get just right. Sounds like this versus this. Great. Okay, so is this really needed, this kind of exposure? Maybe all birds have exactly the same song, they're all converging in the same place. Well, if you never plug this speaker in, they just have this bird sitting here with no sound coming out, then they go into very wild places, these kind of isolate songs, feral songs, where they'll often stutter syllables for tens of seconds, and the syllables are really long, kind of crazy, formless things that sound almost like a goose. Fine. So they need that exposure, they need that practice, and with practice and effort, they end up with a single kind of one-trick pony trick, which is their courtship song. The second advantage of this system is we kind of know exactly where all the interesting bits are. There are circuits that allow these animals to learn, circuits that allow these animals to sing the song in a kind of hierarchical way. And over the last many years, we've attacked this on many fronts. From recent papers in transcriptomics, looking at cell types, we found 8 different inhibitory interneurons, for example, that we think are all doing interesting things here. Connectomics, collaborating with Jürgen Kornfeld and Winfried Denk on those efforts, and we're expanding those efforts now. To photon imaging, even in head-fixed birds, behavioral work, et cetera, et cetera. And so it gives us a real opportunity to take that simple act and take it apart and understand how this works. And then at a certain point, we think, well, this is great. We can look at this natural behavior and really see how a brain can do this. But there are specific differences between what the zebrafinch does and human speech. And anyone who's had maybe one of these animals as a pet knows how flexible the parrot behavior is. Their incredible vocal abilities really put them in this place where I guess in the early 90s there was one parrot named Puck who could speak 1,700 English words, and it was of this species, and I think that's the record for animals that can speak human words. They're incredibly flexible and incredibly gifted mimics. We decided to look more closely at their brains. And so this is just an example of what a very specially trained bird can do. We haven't really taught these animals any kind of words yet. We're really looking at their natural behaviors, but this is a parrot named Disco.
Yeah, come on over. Come on out.
Having captured his owner's attention, he can practice another skill.
It's not easy. easy being green.
It's not easy being green. It's not easy being green.
So if we look at this in the lab, this is kind of what we're seeing when they're using their own natural language, if you like. A zebra finch, I like this movie. It was the first piece of data when I started my lab that we decided to take these this polarized glass, and use that as a way to kind of reveal the female that could elicit the song. So when this current goes across there, the glass goes clear, he sees a female and begins to sing, and you can hear this very repetitive nature of the zebra finch courtship song. In fact, she kind of yawned. I've seen that video enough. She's yawning at the end. He's not doing the magic. Anyway, so this is what just standard budgie interactions are, vocal interactions, and you can just see how rich they are. These are all males, by the way. Excellent. So it's worth pointing out that these two animals, parrots and perching songbirds, probably share a common ancestor, and their brains are made up of these kind of nuclear structures that drive the song. And so it gets very easy as we're getting close to the periphery. They have a vocal organ, in both cases, called the syrinx. There are some differences across this, but for example, this is really wired for each side. So there's a kind of left and right song system that then wires the left and right syrinx, and this is bilaterally wired. But really, they're both vocal organs. There's a group of motor neurons, hypoglossal neurons that drive this muscle structure, and then there's a ball of forebrain neurons that come down and talk to those motor neurons. There's a lot of other nuclei going on, and I'll really feature one at the end of this story. But for now, these things look particularly similar. They have different names. So this is called the RA. Over here in this animal, this is called the AAC. These guys don't have an AAC. These guys don't have an RA. They're separate, but they have very different behaviors. And so we wanted to ask, what are the neural control strategies for these two very distinct vocal behaviors? And so we've done a lot of high-density recordings in both animals now, and this is what a recording in a zebra finch looks like. And this is the efforts of Margot Elmaleh. She's working at the Gilles Laurent's lab in Frankfurt right now as her postdoc, looking at cephalopods. And when she was a student, Margot could record, in this case, 60 neurons simultaneously in that RA structure, that forebrain structure of the zebra finch, as they sing the same thing over and over and over, like that. And you see this kind of QR code that exists for each time that they sing their song. And so we can look at it in a kind of apples to apples comparison here by just looking at this subset of neurons across three different song renditions, and you see how beautifully stereotyped those commands are at the level of spiking. Okay, and so this highly structured song is formed by a repeated series of forebrain commands. So we can take a step back and look at this and see very simple things. So if the animal is quiet, we see just these single spikes. We don't see high frequency bursts, and so but when they do sing, there's this clear, very precise pattern of bursts. And so because no one had recorded from the parrot, the singing parrot, we wanted to ask simple questions like, do we see the same kind of motor activity in the parrot brain as the parrot produces his song? The simple answer is yes. So when the bird is quiet, again, there are single spikes here. But if you look at the firing rate during vocalization, it's higher than it is during quiet. And of course, there's more bursting as well. When we play back the song, we wanted to ask, is this a sensory structure as well? We don't see that. We see just simply that this is motor in origin. Fine. Now we wanted to find out, what's going on with these kinds of interactions, and it can be quite loud. And in fact, what I'm not showing you here in these cages, this is a cage that we've designed to be a kind of figure 8, if you like, and there's an interaction space here. We typically record from one bird at a time. Here we have 2. And we typically have 4 or 5 birds over on the other side. They only sing if they're in kind of a cocktail party. If you have 2 birds there, even they rarely sing. We need some background noise. We need them to feel like they're back in Australia and they've got thousands of birds all around them and they have something to say. And in fact, it's illegal for us to house these birds singly. They have to be housed in large groups. And so this is really just a demonstration to show how we can individually take the song of one bird and not the entire cacophony of what we see in the cage. So what we can see if we put just a microphone down like this and we can listen to both birds is just a ton of sound here. And this is the same kind of sonogram you saw before, where frequency is on the y-axis and time unfolds like this. But we use these very low-cost, head-mounted piezoelectric microphones. They're fantastic, and they do a fantastic source separation for these animals. And so there, we can take that soundscape and break it down really beautifully into what bird 1 is producing, what bird 2 is producing, and you can see 1 plus 2 equals this interacting pair of the kind of ambient microphone. So fine, now we have an idea of what the brain is doing, we have an idea of what the bird is singing. I'm gonna spare you a few slides here so that we can get on to some newer work at the end. But what we found is that if we look in the zebrafinch RA, every single time slice is represented by a different ensemble of neurons, okay? And this is in a paper that came out earlier this year, if anyone wants to see how we got to this cartoon. But this is a just trust me cartoon, if you like. And so there are enough neurons there driving these 7 vocal muscles that are on the order of tens of thousands of neurons. So they can actually create a unique solution, even if the note is repeated here. And this is just, again, a cartoon. If the animal is singing this E note, 1, 2, 3, they have a unique group of cells that are active for each time within that note. And we think that could be helpful for the learning process. If they want to learn one thing exceptionally well, they need to break it down into these kind of unique barcode time slices, if you like. But the budgie is different, and the budgie actually resembles what we have in our own brains, and that is, if a bird sings an E, they go back to the exact same strategy of E, right? The same neurons represent that note. And then if they go to a different note, a different set of neurons is represented. So there's the idea of reuse at the level of this neural circuit. And we wanted to ask what kind of acoustic features are encoded in this budgie brain. And so the first thing that we noticed is this fairly rare sound. Jesse Goldberg, who's at Cornell, also studies this rare sound he calls the G note. It's this low thing here that typically if they're grooming each other or touching beaks, they make this very kind of low growling sound. So we'll call this the G note. They also make noisy syllables like this and harmonic syllables, which really resemble, and people have, other than us, Tecumseh Fitch and others have referred to these as consonant-like and vowel-like sounds. So they make noisy and harmonic notes like this. And we can make this kind of harmonic index where everything over here is noisy and everything over here is harmonic, etc. So if we go back and take a look at this kind of complex spike pattern, you can see, okay, there are neurons that represent this low frequency sound. There are neurons here for this consonant-like noisy sound, and others that are here for the vowel-like harmonic sounds. Okay. But no one really looks at data that way anymore. We have a lot of neurons, and let's find out what the whole network is doing. And one way of doing that is through dimensionality reduction. We can use a state space model and just take the first 3 principal components of each 20 millisecond time slice like this, and just pull them at different moments anytime this animal is singing. And so we end up with this cloud. And what the cloud is just a series of these neural states across all of the song bouts that this animal makes in one day. So you can see this animal is singing, and this is kind of represents the entirety of the neural activity. But what we can then do is decorate these dots and say, what is that animal singing at these different times? And of course, if this is really representing specific notes, specific types of songs, then those dots, for example, this green dot representing a low frequency note, should really be spatially found within one part of this cluster. If this thing is like the zebrafinch and you have different representations for different notes throughout the song, that green dot should be scattered all throughout this cloud. And what we find is that the green dot is really out here in this kind of accessory, if you like, affiliative cloud that exists in this animal. We also see blue dots and red dots to represent the consonant and vowel sounds here within this state space plot. And then if we look at what's in the middle here, they don't always make pure consonant or vowel sounds. Sometimes it's mixed. And in fact, that forms a band somewhere right in the middle here. This map is universal. Every bird we look at has this exact same strategy and this is one that really quite kept to himself, didn't nestle with other birds and didn't have this kind of affiliative cloud, didn't make that cooing sound. But outside of that, we see noisy, harmonic, and mixed bands every time we've looked across these animals. We're now working with Brent Cooper, who's a collaborator in Texas, to do muscle and respiration to kind of put this more in an articulatory space. But we were amazed to see, for the very first time, this kind of articulator map here in the bird's brain. And so we could see even single trial dynamic properties here. So if an animal goes from harmonic to noisy sounds, you can see that animal going from this part of the cloud over to here as you trace through the state space plot. If you go from noisy to low frequency, you start here and end there, or noisy to harmonic. You can kind of draw your cursor through this instrument, if you like, that the animal is making. So there are real similarities to what others have seen using cruder tools in the human brain. And so this is one of my favorite papers ever from Eddie Chang's lab. And what Eddie has found is this using electrocorticography, these tiny sensors right on the surface of the brain. It's our kind of bread and butter technique as well. But if a person repeats ba, da, ga, every time they see ba, there's a specific spatiotemporal activation of this sensory motor structure in the brain. And so there's a clear map of articulation, which is similar to the map of articulation we see now in a non-human brain for the first time in this parrot's brain. Eddie has also found in this dorsal laryngeal motor cortex an area that seems to represent pitch and frequency. So, of course, if we put emphasis on words, if we change the pitch of words, that can generate a question, that can really change the meaning of sentences using these prosodic cues. And he finds this is an area where the activity changes if a person forcibly changes their pitch, or even if you can stimulate that area, it can drive changes in pitch. And we went back to the idea that, well, parrots are also very sensitive to pitch. And this is one of my favorite, not done by us, behavioral papers, where a person whistles the Mickey Mouse song to a parrot. It's not a budgerigar, it's a different parrot species. And this is what the human sounds like. Pretty good. So we wanted to ask something about frequency. So we can't pull frequency out of these kind of big blobby things, but if we, for example, just take these most harmonic notes, which I've shown you here, estimating the frequency with a white line, and look just there in the brain, and those are represented now in this 2D state space plot with these black lines. These gray ones are the noisier, more consonant-like syllables. And now we can ask the question, what frequency is the bird making as he's singing, and the brain is in these states within this cloud? And what we see is this orderly relationship here between high frequencies, low frequencies, and forming this kind of functional map. And so that to me is an amazing thing. And if we look across birds, we see exactly the same thing, this kind of frequency map in motor space that these animals can produce whatever tone they like by simply activating these neural ensembles. So for those who don't believe state space stuff, I mean, it's believe it or not, it is what it is, but want to see the actual spiking behind all of this, These are raw data for an animal that in this case is making a kind of up and then downward frequency sound. So this is a vowel-like warble. And you can see the reflection of that in this neural pond here, right? So as this goes up and down, you can see a kind of U-shape activation of this ensemble. These down here that are not active are likely going to be the more kind of consonant-like noisy related cells. And we can pull a few examples off here. And what you can see is this guy, number 1, is really active for these lower frequencies, between 1 and 2 kilohertz if you like. You can follow that blue line to see spikes per second by frequency. This one seems to like frequencies that are in the middle, and this one likes frequencies that are high. And so you seem to span the whole range. These are the untuned cells. And so we can kind of create this keyboard, right, of the tuned cells. We take the top half, and we, in each line here, just kind of plot out that blue fitted curve. And so this cell at the top, for example, responds the most to the lower frequencies, as you can see in red, and things come all the way down to these guys that like the highest frequency and everything in between. And so we can take these frequency-tuned neurons and do a fantastic job of decoding what the bird is singing. So with just 5 neurons, this is our guess of what the bird is singing if we have no access to the sound file, is our red curve. The gray curve is what they actually are singing. We can even, with the worst tuned cells, see we don't do so well if we take just 5 of them, but as we do more and more, we do very well. And we can compare this to the zebrafinch. It doesn't matter how much data you have in the zebrafinch, you do a terrible job at decoding. Your R value is just right at 0. But the budgie is nearly perfect once you have taken in all the data. So this is an amazing thing, a great brain-machine interface, if you like. We can just read out what's going on in the brain and have a very good idea of what that bird is producing. And so to kind of close out this published work, like humans, parrots have an exquisite forebrain control over vocalizations. We've uncovered this cortical keyboard in which neurons represent distinct articulatory outcomes. And so now what we wanted to do is find out how that keyboard is played. Right? We think we have some primary motor area that's going to these motor neurons, ultimately to muscle, but that's not very exciting. We want to say, now that a keyboard is there, how does the brain actually engage with this? So the first thing that Andrew Bailey, a new postdoc in the lab, wanted to know is, do the warble vocalizations, these kinds of things here, represent just random notes? Are they just exploring the space? Or are these repeatable vocal gestures, right? Are there categories in what these animals are singing here? And so he decided to do a kind of dimensionality reduction on the sound itself. And what he found are these really beautiful, colorful patches. And I should tell you, we've played with a few different pieces of software, and this one from Jan Clemens is beautiful. And we sent in the data, and they form these really nice patches. And so just as a kind of sanity check, we can pull each one of these patches out and say, do we see similar sonograms within each patch? The answer is decidedly yes. And so we can really see this kind of pink guy. Here we are, green, etc. Each one does a very good job of really describing these different sounds that the animal is making. So if we look one step up and we want to say, how are things programmed in the zebrafinch brain? When you really have no leverage, right? No leeway. You learn one trick, and that trick is what you're stuck with for the 10 years of that zebrafinch's lifespan. They're not a seasonal bird. They learn their song in 4 months, and they keep that song really forever. And the strategy is what is found here in HVC. So this very kind of large glutamatergic projection down is this beautiful graph that got me into recording from the zebrafinch brain. So this was discovered by Richard Hanloser many years ago in Michale Fee's lab. This is what made me want to learn more about the zebrafinch system. I thought, oh, we have this thing cornered. It's really tractable. Of course, we're still trying to figure out how all of this works. But what Richard found is that, for example, this green neuron, every time the animal sings this thing, that green neuron is active at just one moment during the song. Pink neuron is active at a different moment. And together, there's about 20,000 neurons that seem to carpet all of the song. So if we look in this same structure, the same kind of equivalent structure, if you like, in the parrot brain, that's called NLC. That's what's actually playing on this piano region of the brain. And what we see is things at first blush are quite similar. What I haven't shown you in the other thing is interneurons are kind of noisy. That's true here too. But if we focus just on the excitatory cells, of which about 90% of these guys appear to be excitatory neurons, they're bursting when the animal is singing, and they're completely off when the animal is not singing. Okay, that's cool. And they seem to be bursting sparsely. Okay, this is also interesting. But what seems to happen, though, is that sequences can be made. In this case, if a person is singing this, if a bird is singing this, and I'm going to call it a token. I don't know what that thing is. They're making a thing. And for now, we'll call it a token for sake of just kind of giving it a temporary name. And we can organize the bursting in this structure for this token and see that it forms this really beautiful sequence. And if we look of these same neurons, while it makes other tokens, other sounds, we don't see any hint of a sequence, right? So it seems that this area that's playing the vocal piano is really revealing this distinct solution to create this behavior, okay? So neurons are reused. Each neuron can be part of different other tokens, but not in this clear sequential way where it's actually driving the behavior. So then we can organize by token 2, and you can see there's no residual pattern in 1 and 3, and we can organize by 3, and there's no residual pattern in 1 and 2. And if we decide to say, that's fine, is this giving us some kind of structure that's repeatable if this animal sings that same token many times? And the answer is yes. And so we could look at, for example, these four example tokens now over trials, and they form these really beautiful sequences every single time. And so now what we have is this kind of ability to sing hundreds of words. And the way that that works is this NLC is sending a command to the keyboard-like region, AAC, and just saying, play this. And then what comes out is this token. So then we performed what I refer to here as neuroacoustic segmentation. We take Jan Clemens's code, we take all of the groups of sounds that come from that code, and now we add as an additional variable the sequences that we have recorded from the brain. And when we do that, it cleans it up even more. And now in just 6 hours, we find instead of 94 tokens, which we get with only audio, this now subdivides and becomes 138, using the neural to kind of continue to split these guys apart into this kind of word cloud, if you like. And I should mention about words that if you look across languages, there's a very simple frequency distribution. It forms this power law, known as Zipf's law. So if it's hard to read back there, this is Hebrew, Turkish, Hungarian, etc. And this is just in the frequency of each word. There are some incredibly frequent words and those that are used very infrequently, and it follows this really beautiful distribution. And we wanted to ask, recently there was a beautiful paper in Science where they just looked at some behavioral measurements from whales and saw that it formed something that appeared like Zipf's law as well. So we decided to take our very advantageous position of having not only the neural sequences that we recorded, but our new kind of neuroacoustic distribution, and ask, does it meet this distribution? And indeed it does. So it forms this kind of power law distribution of use within this bird word cloud. Okay, so in wrapping this part up, like humans, parrots can generate distinct tokens or words that follow a power law distribution of vocal use. We've uncovered network activity in a dedicated premotor structure that generates unique sequences for each utterance. This kind of thing. And so we're going a lot of places with this. The story I've told you with NLC is about a month old, and we're kind of at the point of writing this up. But I think in looking forward, we'd love to know what these tokens mean. Can we speak budgerigar? Rather than just say, it's not easy being green, let's find out what the birds are actually talking about. And we'd also like to know, do pairs of birds have specific conversations? So if I look at the word cloud talking to Ian versus anyone else, how does this differ? Is there some kind of information really being transferred here? And then how do tokens change with experience, kind of plastic properties? And so for grounding, we've put a tiny camera on the head of these birds to see what they're looking at as they're saying different things. This is just a very early movie from there. There's a smudge there, so try not to be too distracted by that. But this is just a few seconds where the animal looks at a toy, looks at another bird, and then ends up looking at and pecking at his own image in a mirror. So there he is. And so we're working with Dan Yeamans at Stanford, who's a kind of machine learning person, and he's asked for 1 million tokens where we have the vocalization and everything that these animals see during this time. We also are recording from above. And what we find in looking from above, if we're using animal tracking methods, is that these animals often really direct their song towards other animals in the cage. And so here's a case where this guy's pointed at this yellow animal. We're just taking these two pieces of our tracking and to indicate head direction. We can see this animal directed towards yellow, purple, and blue. And again, trying to pull out from that, do they use different words in these different cases? And this is right at the outset of our efforts, but it's something that we want to go into moving forward. And then finally, if we plot the data, this is kind of an older plot before we used Jan's code. This was using a variational autoencoder, but I think you can get the point that each dot here is a sound. And if we have two birds that occupy the same cage, you can see how similar this kind of pile of dots are in this case. These are in different cages, and this is similar kind of features. So there's a hook-like thing here that simply is in a different position, islands of sound that are all, if you overlap them here, quite the same in green and red and very different in the blue. And what we can do is image NLC. It's just above the ear, right on the surface of the brain, using a head-mounted 2-photon microscope, and see how those ensembles change over time as they learn new tokens and new sequences. So this is really the work of two people. Zetian Yang was the first author of a 2-author paper. He's an amazing person who's just been with me for a couple of years, but spearheaded the entire parrot effort. And Andrew Bailey is, I don't know, I can't come up with a word for him. He's an incredible person who's really going forward with this, looking at words and sequences and pushing this forward. Thank you.
Thank you very much, Michael, for this beautiful talk. And are there any questions?
So just to clarify, so this keyboard sort of metaphor you have, and you talked about how you can play the keyboard, but when the bird's listening to songs, is the keyboard also playing, or is how does
So what I find is that when they're listening, the keyboard is not activated, but there are moments, and actually we're working with Elad Schneidman on this. So he created an HMM to kind of classify behavior, and he found what he referred to as 2 quiet states. So the animal will often be engaged with another animal, going back and forth, and then he'll hop over and be by himself for a bit. And he's actively thinking about the words, actively thinking about these. So what I mean by that is totally quiet, there's no sound in the cage, and the neural activity kind of replaying these songs is incredibly intense. And so what we want to do, because we can actually decode these really well, right, is ask, are those tokens that he's thinking about things that were really relevant in the conversation he just had, or are they going to be something that he's gearing up to do in his next conversation? And we're doing this now. It's really quite a thing. They're breathing like this. They're in a kind of quiet state, and you see them pause, and then they immediately fire off a train of notes, and then they go back to normal breathing again.
And this is the sort of train of notes that would normally be associated with actually producing the notes,
yes. But there's no sound coming out.
Yeah.
That's right.
And then what region is then, I mean, I'm thinking about a sort of mirror neuron type system. What region is involved in listening to the sounds of others? And because I would have expected it would actually be an overlap, you know.
And in humans there is, right? Yeah. So in these kind of motor areas, there are definitely kind of mirror-looking neurons, and actually Eddie Cheng has recently shown that even at the level of single neurons. So that could represent a difference between these guys and humans. But somehow they have to store these items, right? These tokens, and how they store that, how they use sensory to kind of put these in a storehouse, and how they select the correct token for the correct time is absolutely the holy grail for us. I think that's where a lot of things break down in Alzheimer's, and everything is just, they call it lexical access, finding the right word, right? I can't find the word for this thing. And that's I think, something we can actually study here.
And the zip for just, do you find that the very common vocalizations are simpler? I mean, in human language,
we're looking at that right now. Yeah, so I talked to...
There's a relationship between the complexity
That's right,
and the
that's right. Yeah, so there's a lot of yeah, in human, really commonly used sounds. We are looking at it now. David Popple is someone who's chatting with us about this stuff, and that was the first question he had for us. It's a great question. We haven't gotten there yet. Yeah.
Thank you.
Yeah, amazing talk. So this is could be really naive, but kind of remind me a little bit of Alison Barker that was speaking here about the similarity between different groups of rats.
She calls it a soft chirp, I guess, and there's going to be some familiar interactions.
Yes, the familiar groups would use similar tones, and I was wondering whether, I mean, it does make sense. We do that too. We copy as well. I guess it relates also to what Ian was saying. But I wonder how this scales with group size, because when group size is large, then you actually want to have some kind of differentiation. But yeah, so
I think group size is large. When I see these guys singing, if I were to go back to, I mean, it's clearly evident here, right, where. Who is the gray bird singing to? I mean, not the blue one. And so even with large groups, we find, okay, buddy, we find that they often single out a single bird, right? They become very focused on one, and then they go to another one and become very focused on that other bird. And so there, what we just want to know, and it's very early days, but it appears to be a very different word cloud if they're focused on bird A versus bird B. And what we really want to know now, and we've seen this when we have both birds mic'd up, where we'll have a bird end with, token A, B, C, and the other bird will start with A, B, C, and then keep going from there. So it seems to be this kind of really exciting exchange happening, and we're just getting there now. I mean, it's very, very new. Oh, yeah.
It's a quick question because you had that movie. So this picking at the beak, is that stimulating the other to respond? What you said before, that
they're incredibly orofacial. So there's a lot of mock regurgitation, a lot of beak tapping, a lot of head nuzzling, and we thought that would matter a lot. And so we have these cages that are these kind of, like here. That look like this kind of semicircle here. And we've tried the simple experiment of having a white piece of kind of acrylic in between the two so they can't see each other. And when that happens, they're just hanging out on other sides of the cage. It doesn't matter who's singing. It's as if there's not a bird there. And then if we try to do no barrier at all, they're constantly touching beaks and singing with each other. But simply put, though, I was amazed by this. If we don't let them touch and just let them see the other one, it's equivalent to them touching. They're incredibly engaged simply by vision. They know there's another bird there, and they just focus in on that other bird and go back and forth like this. So they're incredibly visual. But without that visual cue, it's as if the other bird isn't there. And we can track them, and they're just, wandering around and totally bored. Yeah.
I was wondering about one of the slides at the very end where you were showing the sound latent space of birds in similar cages as opposed to ones in different cages. Mm-hmm. Are you planning, or have you looked at how this sound space might transform for an individual bird that changes its environment? If there might be some persistence of the structure over time, or if it...
Yeah, so we haven't done that yet. If we, for example, take this bird and move it to a different place, they'll have a dialect, right? They'll have an accent, if you like. Typically, birds find some kind of solution in the middle, and that ends up being their communication stream. But there are a lot of really fascinating questions. I doubt that it's kind of migrating there. I think there'll be probably, tokens that emerge, and those tokens are and then more and more like this, kind of more in a binary sense, as opposed to something that looks like a zebrafinch is kind of growing there over all of these trials. I think they learn something and can repeat it. But we'll see. We're playing these games now. Yeah.
But this difference in dialects, is this social or just purely physical features of the environment?
It is something that is plastic. It takes about 2 or so weeks for them to really converge like this. And so it's not like they go into the cage and they immediately kind of do this, right? There is some convergence process, and one that we're really excited to kind of follow the dynamics of, but we haven't done that yet.
Thanks. Hi. Thanks a lot. Really cool talk. I'm just not super familiar with the system, but I was wondering if you could talk a bit about whether or not these birds use referential signals, or if they understand referential signals, or is this all just kind of pragmatic communication for social bonding?
We're really trying to get there with the camera. And so the idea there is to say, if this sound is actually rooted in an action or an object in the environment. So we have toys everywhere. This is a kind of silly movie that I played here, but we have a ton of different foods, a ton of different vocal partners, and we're just asking, can we relate even 10% of these tokens to something in the environment? And I think if we just take enough data and throw machine learning at it, it will find patterns. I am trusting in that process. Yeah.
I think it'll be really interesting if you can find convergence like this on a referential signal. I think that was Agreed.
We have a question in the chat.
Oh, good.
Yeah, there's a question from the online audience. Beautiful talk. Sorry for not showing up in person. I am at home sick. About the example where the parrot sings the Mickey Mouse march versus the human performance. I'm curious on whether that was a single trial learning, or has the bird been exposed to it multiple times? I had the clear impression that one of the notes in the human performance was slightly flat, but the parrot's performance was more precise. Could it be that the parrot has generalized a previously learned system, i.e. equal temperament to hit the right note without being off as the human? I'm sorry, no. Let me read that again.
I get the gist of the question. That was not a one-shot learning. So they did have multiple exposures there. So maybe the human did better in a previous iteration, and then the parrot knew how to take the right tone. My former postdoc, Daniela Valentin, who's in Martin's Reed now, or Savizan, I guess, is doing work with nightingales, and there they can just do tone matching, and it's amazing. And so, I think it's something different than that requires some more exposure, but I'm happy to send that paper along to whoever is interested. It's not our paper, it's not our work, but I think it's a really strong example of what parrots can do.
Are you planning to look at communication in decision-making context, like, making them collaborate to solve a task, so if they're smart enough, or things like this?
Yeah. No. I think that what I am interested in with communication can also be illustrated by another experiment, which is we have a robotics expert joining us this summer, and he's building a few plastic birds. We've put these plastic birds in with these guys. They move their wings, they move their head. The birds take them in as their own. They can kind of groom the plastic birds and fall asleep next to the plastic birds. And we can make them sing whatever we want. And so then if we actually, send in a series of tokens that are non-overlapping so that the blue bird and the red bird are singing different things, does the actual bird know, when I talk to my robot friend, I need to use this line of communication, if you like. How they use that to solve a problem, haven't thought about it yet. I think it's exciting. Communication that's a real advantage, right? We have communication, we can build buildings now, right? And I think it's exciting, but it's 2-3 steps down the road. Great question. Thank you all. Oh, yeah.
Yeah, it's really related to that. I was thinking about either using a holographic display or a robotic budgie. Like, what would happen if it wasn't reciprocating? For example, you're trying to communicate with this animal. Does it need to have the attention of the other animal to continue? If the other animal were to say turn away, does it stop, or does it try to get its attention? Like how
it will definitely try to get its attention. I've stared at hours of videos, and it seems to be, it really needs that attention from the other bird, right? So it's foraging for that attention. Once it gets it,
that's why I thought a robot would be very helpful, or a holographic display, so you could actually disassociate things that are normally associated with real birds, so you can look at cause and effect in terms of the reciprocal nature of the interaction.
Yeah, I love
it. Relates to.
So at least with the robot bird, what we're hoping to build into it is some kind of a motion tracker too, so that it needs a bird to be kind of in front of it in order to start with this, and won't do it unless there's something blocking some whatever laser or so.
But really interesting to sort of build up relationships in a closed loop way with,
I totally agree.
Robotic or virtual individuals, so you can sort of see how do pair bond, or how do individuals become associated, because as your intuition, I think it's probably true that they need this reciprocal.
Yeah. I love the idea, and I came to the right place to start thinking about exactly this stuff. So, yeah.
Well, thank you so much again,
Thank you all
Michael.
for your attention.
That was really beautiful, and thanks to you all for the interesting questions. And we have one more seminar talk coming up next week. Looking forward to seeing you around, and have a nice afternoon. Thank you.